So today is my pleasure to introduce Dr. Jessica Spear. Dr. Spear completed her Bachelor's of Science degree in Chemistry and Applied Mathematics at the Western Carolina University in 2010. She completed her PhD in Chemistry from Texas A&M University College Station in 2015, where she primarily worked with AFM analysis of thin films and 2D materials. Following her graduation, she worked from 2016 for 2 years at the EPA, Environmental Protection Agency in Athens, Georgia. Her primary role in that institution, government institution, was research involving AFM of nanoparticle live cell interactions. Since 2018, Jessica has joined the MRL as a research scientist, and she works in the scanning microscopy, scanning probe microscopy core of the facilities. She is one of the responsible or co-responsible for the AFM systems, nanomechanics, tribal indentation tools in the facilities. So with that, thank you very much, Jessica, for putting together this brand new presentation for us. Thank you for taking the time to stay with us here to do this presentation. So please go ahead.
Okay, thank you, Mauro, for that very nice introduction, and thank you everyone for coming to the webinar today. So today I'm going to talk about nanomechanics with AFM. So here you can see an outline of my talk for today. I've structured it this way for a couple of reasons. One is that over the years, I've noticed that a lot of researchers new to AFM in this field are really only aware of it for its imaging capabilities. So today I want to kind of show you the added value of force measurements. And the other is that I want you to not only be aware of what kind of measurements you can make, but have some idea of how it can apply to your own research. So with that, I've tried to show a variety of examples where I can discuss the practical aspects of setting up these unique experiments today. Okay, so to get started, a little about what is AFM. So AFM as an imaging technique is relatively young compared to optical or electron microscopy, but it is complementary in its ability to kind of look at almost any kind of sample regardless of transparency, conductivity, hardness, and even in liquid environments. Not only can we get 3D topographical information, but in some cases also atomic resolution. And over the years, a lot of new modes have been developed to let us measure all kinds of sample properties, and so that makes it applicable to many different fields. So how does an AFM work? This diagram might look a little complicated, but there's really only 4 key elements that you need to be aware of. So the first is a sharp probe. So this will enable you to get those high resolution images. The other is optical detection setup, a feedback system, and a piezoelectric scanner. And what these elements allow us to do is move the sample very precisely, detect small variations in height across the surface, and carefully control the force that we're imaging with. So for today, I want to focus on the nanomechanics aspects of AFM, and so I'm going to focus on these 3 topics today, looking at basic force curves, how to use a force volume technique, and also force mapping, which is the dynamic and sort of higher resolution version of force volume. And so I'm calling these the classic modes just because there's a lot of other advanced modes out there that you can go and check out. But today we'll be focusing on these, and they are both useful for beginner and advanced researchers, I think. So it should be a good example of how to get started in this field. So what is a force curve, and what are all these forces that we're talking about? So here you can see a cartoon of what would be happening if you take this measurement. So the tip would start above the sample surface, and so you would see 0 force in your baseline here. And then you'll bring the tip closer to your sample. As it approaches the surface, it'll begin to feel some surface forces and then eventually make contact, which would be this part of the curve here in the contact region. At a set trigger amount of force or distance that the tip travels, it'll then begin to retract away. And this would be the part where you can see some pull-off forces or adhesion. And eventually the bending of the cantilever will overcome that and snap back to the 0 force baseline. So it's a very simple measurement. What kind of forces can we measure with this? You can see Coulombic forces, even the weaker van der Waals forces can be measured. In air, we have very strong capillary forces, which we'll talk more about later. But I also want to note that if you have charges on your tip or sample, these measurements are also sensitive to that. And the really cool thing is that all these interactions are dependent upon surface chemistry, the type of material, and the measurement environment. So this gives us a way to control the experiment and specifically study interactions between our tip and sample that we are curious about. So before you begin any of these experiments on the AFM, there's a couple of considerations that you need to make. The first one is your sample prep. So it's very important for force measurements that you use a clean substrate. And this is because if you have some kind of contamination on your surface, it can obscure where your real sample is, and you're trying to figure out where to put your tip to take these measurements. The other is that contamination can cross-contaminate your tip, and that can also affect your measurements. So how would you clean it? There's many ways to do that. I've listed a few here. You can use UV light. And what that does is you can generate ozone. It'll break down hydrocarbon contamination from the air into smaller gaseous compounds, and they'll desorb. So it's a non-destructive technique. You can see an example of that here, where a silicon wafer has just been, sitting out in the air for a long time. If you put a drop of water on it, you can see it's so covered in those hydrocarbons that it's hydrophobic. After the UV ozone treatment, you can see that the silicon surface is now exposed and clean, and the water is more hydrophilic as it should be. Or the water is hydrophilic, but the surface is clean. So you can also use a dilute surfactant solution. So this might be something you can do if you have large particle contamination. You can sonicate the wafer in this and then rinse it off. If you plan on doing some kind of surface chemistry afterwards, a basic prana is a good choice because it will remove both the contamination particles, and it will also leave your surface hydroxylated and ready for any chemistry steps afterwards. Another choice you might want to think about is what type of substrate that you're going to use. And so all of these listed here they're really good. You can use them for AFM. They're smooth and flat. But my only comment about this is if you do not need to use one of the transparent substrates, it will save you a lot of trouble. If you would use something like silicon, you'll get a lot better optical contrast, which will help you with your experiment setup. And then the last thing you want to consider when you're preparing your sample and substrate is make sure you're securely mounting them so that you don't get a lot of drift during these measurements. Okay, so your sample is ready. You next want to consider what kind of cantilever that you want to use to take these measurements. So if you go online to a catalog these days, there is hundreds of AFM tips out there. And so it can be a little overwhelming. So I have a few comments about how to choose an AFM tip. You might see rectangular versus triangular tips, and you can take force measurements with either type of these, that's fine. Coated cantilevers, pretty much any coating is better than no coating in general, so just make sure you have a coated cantilever so that you have really strong signal intensity for sensitive measurements, so that's really important. And you might also see options for different tip materials. In general, a lot of tips are made out of silicon, so that's also a good choice. It's good for most measurements. For specific cases, if you want to take a conductive measurement, you might choose a metallic tip. And then if you have really hard samples and you're going to be taking a lot of these force measurements, a diamond-like carbon tip is much harder, so it's more resistant to tip wear, and so will reduce some of that artifact in your data. Spring constant, if you're interested in kind of these modulus measurements that we'll talk about, you want to choose something similar to your sample modulus. And if you're not sure, start with a softer cantilever. A lot of these tips you can buy now have more than one cantilever on the chip, and so you could start with a softer one, and if that's not working, then you can just easily move to the other. So definitely take advantage of that as well. But in general, I've found that a lot of these soft materials, less than 1 Newton per meter spring constant seems to work pretty well. Okay, So after you have your sample and your tip chosen, you are ready to start your experiments, but the first thing you want to do is perform some calibrations on your cantilever, and that's because we want to take some quantitative force measurements. So we want to look at how do we measure the spring constant of the cantilever, and so we use something called the Sater method. The way you do that is you'll start by taking a thermal spectrum. And so here we're going to focus in on this first order resonance peak. And we want to fit that peak because we need a couple of numbers. You need the resonance, and you can see this is a contact mode cantilever, so the resonance is quite low, much lower than a tapping mode. And we also need the quality factor. And then if you have a modern AFM, you know, these calculations can be done automatically for you, but you can also go online and you can perform this calculation yourself. And I think this is really great. If you just Google Sater method, you can see one of the first links would bring this website up. They also have links to the articles, so you can read about the physics and the theory behind how these calculations are being performed. And then you input your frequency, your quality factor, and then you also need to know a little bit about your cantilever, such as the length and width, and then you can calculate it. And one thing to note is that when you do calculate a spring constant, just do a quick 2-second check that it falls within the expected range so that you know that everything went okay with your calibration. Okay, so spring constant is not the only number you're going to need. You need to know how sensitive your cantilever is. So we need to know the deflection sensitivity because, you know, a very stiff cantilever is not going to be very sensitive to small changes in forces on the soft side. So that is very important, and it determines which, it depends on which type of cantilever you're using. So how can you measure that? If it's not done automatically on a modern AFM, you can do this yourself. So you could take a clean substrate that's really hard, such as silicon. You can take a force curve on it, and then we basically look at this in raw deflection volts and distance, and then you can take the slope of this contact part of the curve here. And the reason for that is because that silicon is so hard, we know there's no indentation happening, so all of this signal is coming from the bending of your cantilever, and so we can get a good sensitivity factor from that. Now you have a spring constant, you have this deflection sensitivity. You can multiply these by the set point voltage that you're using, and you'll know approximately how much force you're applying in a meaningful way to your sample. So there's one more calibration that you need to be aware of. And so if you're interested in quantitative adhesion or modulus measurements, you also want to know what is the radius of your tip that you're using. And so there's a few ways out there to do this. You can see you could use direct probe imaging approach where you can use an SEM. You can see a colloidal probe here. With this method, you're probably going to need to use some kind of image processing software to extract out any numbers about the radius from that. And you also need access to an SEM and someone who knows how to use it, so there's a bit of a barrier to that. Another common method is blind tip reconstruction. And so how you can do this is you can use a substrate such as this NIO probe, which is great for that. It has tiny, sharp spikes all over the surface, and they're oriented in a random orientation. So when you scan it, you'll get a really good convoluted image of your tip on the surface. And then you need to choose a software that can use these complex algorithms to basically extract out the tip from that image. And so this is a bit of a learning curve because you need access to this kind of sophisticated software. There may be some free ones in the literature for like MATLAB programs, but you also need to know how to use it, how to input the parameters and interpret that correctly. So there's a little bit of a learning curve barrier on that. The third method, which I think is the easiest one, is reverse probe imaging. And so you could use a grating such as this test grating here, which has a lot of tip-like spikes all over the surface. And the way this works is you'll scan the surface, and instead of getting necessarily the image of your spike, if your tip is blunter than the spike, then you'll see the image of the tip. And to kind of help you understand that, I'll show you an image using this technique with a colloidal probe, because the colloidal probe is much, much larger than the spikes. When you scan across them, you can see the image of the probe instead. And so then all you need is to take a cross section, and you can go in and look at the radius of curvature of your probe, and you can use follow like the paper did here, take a rough estimate based on the height and width, or you could use some free fitting software and just kind of fit this to the radius of curvature, and then you already have your number. So this is very simple. The grading that you use for this is really cheap, and so you can do this on any AFM, and it seems to be the easiest approach. There's a few other considerations if you decide to use this. It's also great for checking your general tip quality, and also identifying if your probe has any kind of contamination. So this is an image I took of a brand new colloidal probe that I had bought from a company straight out of the box, and I checked it before I did my experiment, and you can see there's tiny contaminant particles all over that you wouldn't even have been aware of if you hadn't checked it first. And so it's really great for that. And then in some cases, if you check your tip before and after an experiment, afterwards you may see that some of the sample has cross-contaminated your colloidal probe, and you can see that in the image, and then it can also sometimes scrape that away. So it kind of has that little bit of benefit. And just remember that it's good for the blunt or large probe radiuses, but this is kind of the probes you're going to be using anyway, so it should be fine. Okay, so those are all the calibrations that I want to talk about. Now let's look at some measurements, and I want to start by looking at adhesion. So in this case, we'll look at a functionalized AFM tip to kind of measure the chemical interaction between the tip and sample. In the literature, you'll see this technique called chemical force microscopy. And it's been around for a while. So since the 90s, people were looking at just fundamental interactions between different functional groups on the surface, and even more modern examples where you can take a tip functionalized with biomolecules such as DNA, and then you can study site-specific binding on your surface. But what's really cool is that it's the same simple method, and it's still just as relevant, still just as powerful as ever to get these really good measurements. We're not only getting the nanoscale spatial resolution with our adhesion measurements, but also we can do this in native environmental conditions. So if you wanted to functionalize a tip, how would you do it? It's really easy to do. Most AFM tips are made out of silicon, so you can use silane chemistry. You can see an example here. And so you'll have a silicon group, and it'll be attached to either some chlorosilanes or methoxysilanes, and then pretty much you can use any organic group that you are interested in here. And so if you go to a company website like Gilleste, they make hundreds of these molecules pre-made, very cheap, so you don't even have to do the synthesis yourself. And then how would you put this on your tip? There's a lot of deposition techniques out there. One simple way is just to use a liquid phase deposition. So you need to choose, based on the molecule that you're interested in, an appropriate alcohol or solvent, organic solvent that it's soluble in. And then you just take a small amount of that and just soak your AFM tip in it and give it time, because this is a self-assembled process. So it will just go through these different steps here. It'll react with a small amount of water that's present in your solvent. That'll initiate the reaction, and then it'll condense to your surface, and it will start ordering. So it'll have some hydrogen bonding, some bond formation to the tip, and so you just give it time. But also, you may get, as a side product, more than one layer on the surface. So for this technique, it's really important that you have the correct solvent to rinse away excess loosely bound molecules. We don't want those floating around when we do the experiment. Okay, and so one of the questions I get when people go to do this is, well, how do I know that this reaction worked on my tip? And so one way I might recommend is you have silicon tip. It's the same chemistry as the silicon wafer, so use whatever reaction protocol that you've decided on, test it on a piece of silicon wafer, and then use some kind of chemical characterization technique. Infrared spectroscopy is very common. You can easily check if your functional groups are on the surface. And then if it's applicable, you could use Raman spectroscopy as well. And if you want to do this at MRL, please contact Julio Suarez for that information. So let's look at one example of using an AFM tip to actually measure adhesion on the surface. And this example comes from 2017, and it's on looking at a functionalized tip with different functional groups to a silicon wafer with graphene on it. And so there is unique experimental setup you have to consider here. The graphene is not everywhere on your substrate, okay? We want to put the tip here, so we're going to have to pre-image the sample before we do the measurements. And so that can be a barrier because the silicon wafer has really high friction forces, and that can easily shear off a tiny monolayer of this molecule, which we don't want to happen before we take the measurements. So you need to think about how to preserve your tip quality before you can actually get these measurements done. There's a lot of ways to do that. I've seen some really cool, unique ways from different people. One way, just as an example, is you could just use some simple optical images. And what you're gonna do is you could use kind of this zoomed out optical image. You can kind of get an idea of where your tip is on the cantilever, and then place the cantilever where the graphene is, and then use surrounding area to kind of help you align it to the right spot, and then use this zoomed in optical image to kind of, once you start a scan, quickly determine, am I on the graphene, am I like just to the side of it and I need to move the tip here. This will help you find your sample within like 1 or 2 scans, you know, of course with practice, but it will save you a lot of time searching and scanning, and then you preserve that tip quality in order to get those measurements. And so once you get all these measurements done, you want to do some analysis of the adhesion. And so the first thing you need to do is choose a contact mechanic model that's appropriate. In this case, we're looking at the JKR model because we want to account for the compression of the SAMs within the contact. And so you can use this equation here. And it's very simple. You need to know that adhesion force, which comes from that pull-off region that I showed you earlier. And then you need that calibrated tip radius. And then once you get this, you can plot your data and you can see there are actually differences when you functionalize the tip with different functional groups. You can see this hydroxyl group here is basically a bare silicon tip that's been cleaned, and it has the least amount of adhesion. And then if you use something like this methyl terminated group, it's more inert but a little hydrophobic, so it has more adhesion to the graphene. And of course, if you add this phenyl group, you get increased pi-pi interactions, and then you have even more adhesion. So it's not an in-depth explanation of what's going on. You can read the paper, but it shows you the power of this simple technique, just functionalizing your tip, and you can get a really good understanding of how your sample is interacting with different surface chemistries on your tip. So what about if you have topography on your surface? So for comparison, let's look at graphene oxide on a silicon substrate. And the only difference is that the substrate has some roughness. So in this case, the chemistry is not being varied, it's just the roughness. So when the chemistry doesn't change, you wouldn't really expect the adhesion to change, but I want to talk about the effect of topography so that you understand how artifacts can be involved in your data if you do have changing roughness across different samples. So you can see we looked at a smooth substrate with just a flat graphene, and then a very rough case. And so what you'll see if you take these measurements is that the adhesion is actually changing quite a bit. And you'll see that when you have really, really rough surfaces, the adhesion will decrease. And so the reason for that is 2 parts. One is that we have a change in contact area between the tip and sample. It's different when you have an asperity-asperity contact versus the single asperity on kind of a flat wafer. And the other is that we have these measurements done in air. So we have a capillary force. And that happens because we have humidity and a small thin layer of water at the surface. So when the tip makes contact, it'll form a capillary. And you can see these really cool SEM images. The capillary force is also dependent on many things, such as the amount of humidity in the air, the hydrophobicity of your surface, and so also the roughness. And so now we know, since we held all of these constant except for the roughness, we know that it's the roughness that is actually changing our adhesion and not actually related to chemical adhesion. And so you need to be aware of this. But now that we understand the role that a capillary force plays, we can also use this to help us understand some more complex data, such as an adhesion map. And so we generate adhesion maps with something called force volume. And so we choose, instead of taking a single point, we're going to choose an area here. For example, I'm choosing a 0.5 micron. And then you choose the number of force curves per line, and you make a grid of these. And so you kind of, that's how you can control the resolution. So it's not as high as a regular image, but it's for these large asperities high enough that we can actually see some of the topography structures. And so from this data, you can see two images. We've extracted a height image, and then we've looked at the variation in adhesion across the sample. And you can see in these bright regions, the adhesion is higher, and it's clearly happening around the sides of the asperities, and that's because of that increased contact along the sides. So this is a cool way to map adhesion across a surface, and also now you're aware of these topography artifacts. And we can also do something else from this set of data. We can extract out individual points that we may be interested in. And so we can see here in this region, there's kind of a suspended area of graphene where there's no particle or asperity underneath it. And what we see here is in the retraction, there is a change in adhesion, but it's not really due to the adhesion, it's changing due to this deflection here, this change in slope. And so what we suspected is that this capillary force is actually strong enough to pull the graphene up to the tip as it retracts away from the surface in this pull-off force cycle. And so what we did is we took that same set of data and just did a little more advanced processing, and we extracted a height image from each point along this retract curve. And then what you can see is you can see at the higher forces, the tip is pressing the graphene down and conforming to the topography. So you can see the red parts, which are the higher peaks, are the particles. And then as the tip comes up, you can see actually how the topography is changing. And you can see that at the pull-off force region here, the graphene is actually being lifted up in between these particles. And so this is really powerful. You have one simple set of data. You can see how the adhesion changes across the sample. You can look at how your topography is changing. And so you really get a good understanding of how your tip is interacting with your sample. Okay, so both of these examples we're looking at graphene on a hard silicon wafer. So let's look at what happens if you take these measurements on a soft material such as bacteria. So here we have a case where we were looking at bacteria on different topographies. You can see here it's a spiky substrate, and the researchers noticed that the bacteria was responding different bacteria to the substrates. So we thought, what if the membrane stiffness is impacting this ability to be pierced by the substrate? And so we wanted to develop a force method to kind of probe this and see if there's any differences between the membranes. So the first approach we did was we tested out the same technique that we did on the graphene, which was do a pre-imaging, locate your bacteria, and then perform your measurements. The problem is that the bacteria is pretty large, a couple microns wide, and the tip can easily push it aside when you're imaging. So we tried a wrinkled PDMS substrate to kind of wedge a bacteria between two wrinkles, hold it still. So we imaged, we found one that wasn't scooting away from us, and then we zoomed in on it. So this would be the area we chose for a force volume. And then we collected a force volume. And afterwards, we did one more scan. And this is to make sure that during this measurement, we didn't actually drift away from the sample spot. And so we didn't drift, you can see here, but the bacteria is gone. And that's because we used a fixed bacteria the first time, and it doesn't have any recoverability. When you pierce it with the tip, it basically shreds and then kind of drifts away in the fluid. But this method does work well for the live bacteria. So you can see a really nice set of data we were able to acquire. Here is a bacteria we found, we imaged, and then we did a force volume over this whole area. And you can see extractions of 6 different force plots from this force volume. And so this time we're not looking at the adhesion, and we're in fluid, so there's no adhesion here. You can see on the substrate, there is little indentation. The slope is pretty straight. But here on the bacteria, it's a lot softer. And so we can see this change here. But what we are interested in is when you apply enough force, the tip will actually pierce the membrane, and then we see what's called a breakthrough force, this change in deflection. And you can actually measure the force at which this happens. And so this works pretty great. This is what we wanted to measure. The problem is now that we have this really cool image, but it takes a long time because you have to scan and find one bacteria that's not moving away from the tip. And so this isn't feasible because we really need a large statistical distribution of measurements to get an idea of these differences. because we're looking at really small forces. So we decided a different method for this would be appropriate. So we skipped the imaging because we already have some good images. We don't need them every single time. We went back to a silicon wafer and we populated the silicon with a lot of bacteria so that anywhere we put the tip, there should be some. And then we just straight away took a force volume map over a larger area. You can see this is 5 microns so that it should be able to find one of or 3 of the bacteria. And because the silicon is flat, the only changes in height that you should be seeing is from bacteria. And so these bright regions, they're low resolution, but we know that this is where our data would be coming from. And so now if you not only do it this way, but then you limit the force curve region. So we only are doing a force curve over a small distance, which is small enough to detect the change that we're looking for. Then this actually, with this low resolution, it only takes about 1 minute to collect this data. And so now you can see it's very feasible to collect a lot of data. And so in total, we collected over 50 force volume maps and over 10,000 force curves per bacteria type. So now we can get a really good estimate of what's happening between these different bacterias. And we also know the radius of our tip. So that's how we're controlling that spiky kind of simulated aspect. So we're measuring the force, breakthrough force, and what I want to point out now is that this data looks a lot noisier than the data I showed you before. And that's because as you begin piercing all these bacteria, eventually the tip will become cross-contaminated from these samples. It's just the nature of this type of experiment. We're on purposely, you know, breaking apart the membrane. And so there's nothing we can do. It just becomes part of your sample variation here. And so that's another reason why you should be taking a lot of these measurements. And so you can see one thing I want to point out is that the breakthrough force of these membranes is 1 nanonewton or less. And so especially if you're just trying to image topography, you really, really need to pay attention to the amount of force that you're imaging with, because you don't want to deform the topography or shred the bacteria. You want to actually image it, so you're going to need to keep the force much lower than this. Okay, so this is just looking at one aspect of these soft materials, which is the breakthrough force, which is unique to looking at membranes. Now I want to talk about a more common technique, which is looking at the modulus of soft materials. And so for this, I'm going to use an example of a smart polymer. So here, this is an AFM image of a sample that contains a NiPAM copolymer, and it got a lot of silica particles embedded in it, which makes it very rough. And the purpose for that is to make it super hydrophobic. The problem is now we have hundreds of nanometers of roughness, and once again, even though the sample is soft, the topography does affect the measurement. So in this case, this is now a case where you're not going to use a regular nanoscale sharp AFM tip. Now you want to use a colloidal probe, which is microns big and can average over the roughness over a larger area, reduce that topography artifact. And the other reason is now we have an example where I can show you how you can change the environment to actually affect your measurement. And so you can see some raw data here. This is a silicon wafer, so there's no indentation, and then you can see the slopes changing for the same sample, and all we did was change the environment, such as the salt solution and the type of salt. And you can see that there is some effect on the modulus, but how do we actually calculate the modulus from this type of data? So you're going to want to use something called the Hertz model. And so for this model, it takes into account the elastic modulus of both your tip and your sample, because when you take this measurement, you get the indentation and bending of the cantilever. But since we know that the tip, the modulus of the silicon tip is much greater than the polymer, we can make this assumption and simplify this equation to a more convenient equation to use. And so now we just need to know a few parameters. You need to know the estimate of the Poisson ratio of your polymer. You need to know that calibrated radius, once again, the amount of indentation, which you can get from the force separation curve. And then because if you indent at higher forces, you indent larger, that depends on load, which is the force. So you need to know that calibrated force that you applied during the indentation. And so once you do this, you can get an estimate of the reduced modulus, and you can see that if you cycle through the different environments, you can actually get an idea of how your modulus changes over time. And NiPAM is also a temperature responsive polymer, and so just to let you know, you can do the same thing with temperature, and MRL has a polymer heater, so we can do these measurements in our facility as well. Okay. So if you were thinking, that's a lot of steps to get my modulus, well, you were right. And it's a good time to tell you about our Puma nano-indenter. So this instrument was developed specifically to make that whole process of modulus measurements on soft materials really straightforward and easy. And so even the tip mounting is simplified. You can do it with your hands. You don't even need a tweezer. You can just plug it right in. It still uses a cantilever and a colloidal probe. The main difference is that it's measuring the displacement of the cantilever through interferometry. And so it's going to automatically calculate the Hertz model to your data. And then it has an open stage, which is actually a benefit because now you can easily work in fluid. You can see the open stage here. You could put an entire petri dish with large samples under it. And so you're not limited to sample size anymore. And so one of the most common samples that we get on the Puma is hydrogels. And they have some unique experimental considerations that you got to think about when you're taking these measurements. So here you can see what I was mentioning before, a petri dish, and then you have a sample. You can see your tip because we have a side view camera and a top view camera for this instrument, which makes it very easy to set up. And you can see the tip here. This is a picture of a microgel sample here, and you can see, if you can see it, it's transparent. So that's also one challenge of optically identifying where your sample is located to put the tip on it. Also, you need to make sure that hydrogels are fully hydrated because you don't want them expanding or contracting in fluid during the measurement. And the most important part is making sure that they are immobilized because these guys can float around in the liquid and you can't take a force measurement on a floating sample. So that would be the main experimental challenges for this type of sample. But if you get it all to work, you can see the power of these measurements even for these really soft hydrogel materials. And this, I'm showing you a quick screenshot of the free data analysis that comes with Puma. All you have to do is load your force curve. It will automatically use the Hertz model that you select. You choose the fit range, it fits your data in this red line here. And then it calculates that effective Young's modulus here. And it's based on the probes that's pre-calibrated for you. You don't even have to do that. So the probe you buy, it tells you what the radius is exactly and the probe stiffness. So those parameters are automatically input. And then you can see this is small kilopascal region. So it's very sensitive measurements. And you can download this yourself at this website here for free. Okay, so the last part of my talk, I want to switch gears and talk about force mapping. So this is kind of a dynamic technique. And it uses a sinusoidal driving voltage, which gets applied to the Z actuator. And this is because we're oscillating the cantilever. And now we're not talking about small image oscillations. We're talking about oscillating to the length of a force curve. So that's why it's being applied to the Z actuator. And we're doing this at really low frequencies, such as a few kilohertz, which is intentional because we want it to be far below cantilever resonance. And so what happens is this has an advantage over the other imaging techniques. And so this mode is useful for, of course, mapping the forces like I showed you for adhesion and modulus. And I don't want to show you examples of those again because it's the same thing as force volume except for just a little bit higher resolution. I want to talk about the aspect that is unique to this, and that's really what I think is the most powerful about this is its use as an imaging technique. So compared to like tapping mode in fluid, tapping mode is so sensitive to, you know, the liquid environment and the changes in temperature, and so it makes it unstable. It's a dynamic resonating system in fluid. It's challenging. And also contact mode has these shear forces, so what happens is this mode eliminates both of those problems, and so it can get very precise imaging in fluid with low force control. So it's great. How it works is when you apply the sinusoidal driving voltage, you'll go through a cycle of this force curve. And so you can see the image here. You start away from the surface. You will snap in with some approach curve. And then the feedback is based on this peak force, which is how far the tip will indent, and then it will retract off. The unique part that you'll see in this curve, which you won't see in a regular force curve, is this snap off, which may have some ringing of your cantilever. And you don't need to worry about that. As long as the ringing doesn't carry through to the next cycle and affect your feedback, then it's fine, because we're mostly interested in the modulus measurements or the adhesion usually. so that's fine. So I want to look at an example where this mode can image a sample that wasn't possible by contact mode or tapping mode. And so you can see an example of some nano-pillars here. And this is not ideal for AFM. They are very high aspect ratio, they're very soft and floppy. That could be very challenging to try to image with a tip. And so obviously the SEM images look nicer and that's what you should do. The problem was the researchers found that when they took this sample and they put it in fluid, when they took it out, it had a different structure. And so now the question that we have really needed an answer to is, what did the structure look like in fluid so they can model what's happening with their sample? So we came to the AFM to answer this question, and we used the force mapping. And so we tried in contact mode and tapping, and it did not work at all. So we first then switched to force mapping as an imaging technique, and we confirmed the same images that we saw in SEM. And then when we did this in fluid, you can see we were able to determine that the structure was the same as in air. And so this probably happens during that drying process, and so it's not happening in the fluid. So this is one easy example, and I want to show you a really complex example to really illustrate the power of this technique for, once again, looking at these nanoparticles such as graphene oxide and these thin biomembranes. So this is an example of some work I did at the EPA, where we looked at, they were looking at screening methods for exposures. So if you get exposed to nanoparticles in such as an aquatic environment, what would happen and how can we easily detect that? And so they were using the quartz crystal microbalance for this technique, which uses a sensor, and you flow through solution and it's resonating. And then as the mass deposits, the frequency shifts. So it's all the data is just frequency shifts. And so you don't, it's kind of a blind technique. So in order to understand and interpret the data, what we want to do is take this same setup and mimic this experiment where we could use the AFM to study in situ what's happening during this deposition on a membrane. So can we visualize this in fluid? And so this is also another question of, does it look the same in air as it does in fluid? So if you deposit graphene oxide, you can see it's very common to see wrinkled structures everywhere. And this is a sensor surface here, which you can see a little bit of roughness. And so we wanted to look and compare in fluid. So I want to walk you through the complex nature of doing an in situ measurement like this. So you're going to use the same silicon sensor that the QCM does, because we want to mimic it as much as possible. And then you want to set it up in air first. And so I like to tell people to try this because when you begin a complicated experiment, set up everything step by step so that if you have a problem, you can troubleshoot it at that step. If you go ahead and throw everything together at once, you're not sure where the problem is coming from. So a quick setup in air to check if everything is good, and then you can add fluid. And I'm saying inject filtered fluid because I'm looking at particles being deposited on a surface. I don't want anything floating around, any contamination in that fluid. So you could use something like a syringe filter to just quickly cleanse the fluid before you add it to the AFM. And then, because we're doing this in situ, these bilayers, they don't last in air. So you have to make them, form them in the fluid. So we inject the vesicles, you wait for them to deposit and form the layer, and then you have to flush the cell of all the excess. And then at the next step, you're ready to add your particles, inject them, and then wait for them to deposit. And because we did all this injecting and flushing, now we have to wait for drift, because you can't image if you have a lot of movement happening inside the cell. The liquid cell is just, you're not going to be able to get a stable image. So what happened, just to show you, is that we didn't see any wrinkles at all. And so the power of this force mapping, you can see this really clear image. This is of the bilayer. So we're imaging with less than 1 nanonewton of force in fluid. And you can see we didn't pierce the layer. And actually the graphene looks like a balloon structure with these edge interactions, which has been speculated to have increased edge interactions because they have all those extra dangling bonds. And so this is really cool that we could actually see this, and I've never seen that before. So just to show you how powerful this mode is, now we're getting data that we've never seen before with the AFM. Okay. So I've showed you a variety of examples, and I hope that you feel inspired to try these yourself if you are going to do them at MRL. we have two types of AFM. We have a Cypher and an MFP3D. You can do all these force measurements as well as the colloidal probe measurements on the Puma. If you are interested in force mapping, you need to use the Cypher. And if you want corresponding images to the force measurements, you have to use the AFM. So with that, I hope you come back next week for the Intro to Small Angle X-ray Scattering talk by Juan Lopez. And with that, I'll take any questions.
Thank you very much, Jessica, for this excellent presentation. As I think everyone could see, lots of hands-on experience, a lot of experience in these techniques. Several of the data that was shown here was actually data acquired locally here in our own tools. I guess one question, we have a few questions here. Let me start with the first one here. When you talk about in slide 13 about the clean silicon wafer, the wafer needs to be clean. Can you give a little bit of reference for how clean does the wafer needs to be? Are we talking about, do we actually need to do like some kind of wet chemical cleaning as we do on silicon wafers when you were going to do like thin film deposition or device process? How clean that wafer needs to be? It's slide 13 when you talk about, I think, some calibration.
Yeah, so, you know, as a surface scientist, I'm going to tell you, it needs to be as clean as possible. Okay.
That counts.
Yeah, this is cleaner than like just being sterile in a surgical room. Like you don't physically want anything on your surface to interfere. But you know, it does depend. If you're depositing something like a very thin monolayer, that's going to be very sensitive to even a little bit of contamination. But if you're doing bulk techniques, then probably, you know, just a quick sonication is okay.
Okay.
Yeah.
Good. About slide 35 on the Puma, how deep can the water be for the Puma system? At what point does it actually start affecting and possibly damaging the instrument?
Oh, can you say again?
Slide 35, when you talk about the Puma, there was a question about how much water, how deep the water can be when you're using that instrument. So is there a limit there that at one point you put too much water and start affecting and damaging the instrument? Can you comment on?
Yeah, so how much fluid that you use in the liquid cell on the Puma? One, it would depend on, of course, there is eventually a weight limit that the stage can handle. So the fluid would definitely be the most heavy. And then if you want to work in a droplet like the AFM, you're welcome to do that. I find that very challenging. But for the Petri dish setup, what I like to do is have enough fluid just above the sample that you can get the tip into the fluid, that it breaks the surface tension, and you can actually get a stable optical signal. And once that happens, then that's enough fluid. So other than that, it's just excess. And you can start with more to do this optical signal calibration, and then you can just take some out as you're doing the measurement, so you can change it over time.
Okay. Now, one question about the hydrogels work. How do you immobilize the hydrogels to the substrate?
So that is a very, very good question. We've spent many hours debating this. So if anyone has any ideas, throw them at us. We'll try them and let you know. But so far, I've discussed and we've tried, one method is functionalizing your surface. Like I mentioned, you clean the wafer, prep it for surface chemistry, something that would interact strongly with your hydrogel on the bottom part of the sample to kind of hold it still, increase that affinity to the substrate. Other people maybe mention using rougher substrates, but I don't think that would necessarily hold it down. I've seen some groups that actually mold the hydrogel to the dish that they're going to be taking the measurements in so that it's already kind of stable and pre-hydrated. So those are a couple of methods, and then I've seen some really creative engineered ones where they come up with really unique side pins that can pin down the sample at the edges, and then you take the measurement in the center. So any ideas you have, throw them at me. Those are the few that I'm aware of.
Okay. All right. Now the next question is about a analysis, mechanical test in a fiber material. Say, for example, that you have a fibrous material, let's say electrospun materials with some individual fibers. Can you test? Can you do mechanical tests on these fibers along different directions? Let's say X, Y, and Z axis, like push and pull using AFM in a liquid solution, liquid environment?
Push and pull the fiber with the AFM tip?
Is this a question if you can actually have, I mean, is it feasible like in a AFM, you do push and pull like to test in an individual fiber? I think we came through this question a few.
Well, if it's like protein being attached to your tip, there are pull experiments where they attach it to the tip, and then they as the tip pulls away, they can measure the unfolding of the protein. So, and that's really sensitive measurements. I don't know if that's the same as like a fiber that might be stiffer, and then I'm not sure what you'd be measuring by pulling away with that.
So what I suggest the researcher who put that questions, follow up please with Jessica, and also Kat in the lab. So you have Jessica's, if you put your last slide there, Jessica, people will see your email. So they can contact you directly. One more question here. Can you comment on how to calibrate these tiny forces with the tip? I understand the question here.
So calibrations are pretty much what I showed you in the beginning. If you are aiming to get less than 1 nanonewton of force, that's coming into play not from the calibration, it's coming into choosing the really soft cantilevers who can be sensitive to forces that low without damaging your sample. So basically, like 0.1 newtons per meter, that force, you could apply a lot of force to the sample and it will, not a lot of force, you could bend the cantilever a lot without applying a lot of force. And so that is how you can get a stable image with tiny forces.
Okay. Now, one question about the functionalized tips that you mentioned there. One question here is about how much, when you try to estimate the amount of FM work to be devoted to a certain tech instrument, how much time you should allocate for like the preparation of the tips. I guess the question is that does the researcher supposed to have his own tip functionalized and fully characterized before starting the FM measurement? Do you understand the question?
So should you give yourself enough time to do all the calibrations before the measurement?
Yeah, how do we estimate how long time you're gonna need?
Well, there's different thoughts on how you could approach that. So one is you could give yourself a longer session on the AFM and do all your calibrations the same day. You could do calibrations a different day so that you have like all your tips calibrated, or you could wait until after your experiment and then do the calibrations, because sometimes what I've seen a lot of users, they get very frustrated is they spend all this time calibrating their tip and then the tip breaks and they didn't even get to start the experiment. So they feel like time was wasted. So some cases they might want to just wait till they have a tip where the experiment produced good results and then take the tip and then calibrate it afterwards. But if you need to know radius changes, you might want to check that before and after. But they're quick and easy, so it's however you want to design your experiment.
So what suggestion do you have for doing force measurements of DNA, like a few nanometers thick, immobilized on surfaces?
I guess I would need to discuss with that person what their objective is, what they're trying to measure. I'm not really sure how to answer that because I don't know what they're measuring.
All right, that's another one that I think I recommend the attendee to contact Jessica directly for that. How effective is it tapping molding liquid? What cautions needs to be taken care, if anything at all?
I was really skeptical before I came to MRL. I think, yeah, Kathy actually showed me how to achieve good data with liquid and tapping mode. And so I've kind of changed my mind. I think you can get some good images, except I will tell you, you can't control that force. So if you need force control, I would not recommend tapping in fluid, but it is quite frustrating. I actually prefer contact mode in fluid. We could get into this whole discussion. That's a whole different thing. But yeah, you can do tapping mode in fluid. Just be extra like caution wise, don't spill the liquid, the normal precautions of working in fluid. And then you need extra time because things drift around. And so that's like a whole different discussion we could do later.
Okay, probably one final question here is how to determine the resonance frequency of a cantilevering liquid And the person here says that sometimes I find it the frequency changing all the time as the level of the liquid in the cell is always decreasing.
Yes, that is exactly the reason why companies spent so much time and money developing force mapping, because yes, the resonance frequency will change based on how close you are to the surface, the liquid environment changing, the temperature fluctuations. And if you want to find that resonance frequency, it's usually about, it's about 1/3 of what it is in air. So first you need to look in the smaller frequency region. And if you can take a thermal spectrum, sometimes that can help you determine what is the correct frequency in fluid. But if you can use fast force mapping or peak force tapping, those techniques will avoid all those problems and you'll have a much better time imaging in fluid.
All right, I think that answers the number of questions you had. Again, anyone who has interest in these techniques, feel free to contact Jessica Spear or Kathy Walsh. Both are our experts in the scanning probe microscopy core. Also, don't forget to check, because both of them, Kathy and Jessica, are very active in posting videos, tutorials, tips, and information about how to do difficult measurements in our YouTube channel. I know that Jessica just had 2 recent videos uploaded in that channel as well. So with that, I would like to remind you guys that don't miss next week another brand new tutorial. Dr. Juan Lopez will give an introduction to small angle X-ray scattering. We're looking forward to that next Thursday, noon Central Time. Any more comments? Anything else to add here, Jessica?
No, that's it. Thank you everyone for attending. I hope you come see me at MRL and do these measurements.
There was a question about the availability of your slides in PDF form or so. I suggest everybody to contact Jessica directly.
I'm happy to share the slides. Just send me an email.
There you go. All right, thanks everyone. I see you guys next week. Remember, we'll start training new users on Monday here in the MRL. If you have any questions, contact me, contact the team that is listed over there. I hope to see you guys back Thursday noontime, introduction to SAC by Dr. Juan Lopez. So stay safe and have a great rest of your day. Thank you very much again. Bye-bye.